Thanks for the introduction. Very good pronunciation of a French name. I'm impressed. So we're going to go beyond the voxel as the previous talks and talk about diffusion tractography, really at the basic level to try to get you to understand tractography, the principles and the methods, and not just see tractography as a black box. So I have no conflicts of interests. So I'd like to start with this slide where you can imagine the brain as a bunch of wires connecting short or long distance areas of the brain, and diffusion tractography as a non-invasive way to go about finding these structures and these brain wires. A more realistic view here in a 2D slice, coronal slice, and these are the type of figures that doctors use to learn anatomy. You can appreciate that you can start tracing out connections in these slices and see some of the U fibers, some of the long-range connections that are also called fiber bundles. This is a 2D histological slice and drawing. In 3D, here you can see a postmortem brain dissection where some of the temporal lobes has been removed, and you can appreciate a 3D structure that looks like the optic radiations. So curving near the temporal pole with the Myers loop and projecting back towards the visual cortex. And nowadays with tractography, we can perform what we call virtual dissections. So using a full brain tracking, start dissecting away some of the major fiber bundles or big highways of the brain to try to study white matter neuroanatomy, compare left and right asymmetries, for example, or apply different metrics along these fiber bundles. And just to give you an idea, here in light blue, you have the number of papers per year that have been published in the literature. So in yellow is the standard diffusion imaging papers, which has quickly grown in the 80s, 90s, 2000s, and diffusion tractography from its invention in 1998, the first paper, you can see quickly growing, also following an exponential trend with more than 2,000 papers. And this is even growing even faster these days with new terms like connectomics, tractometry, and different applications of tractography. So the plan of the talk is to review the basic principles of deterministic and probabilistic tracking. You must have heard these terms. You see these terms in papers. I'll try to explain them with simple images. Show some DTI versus Hardy tractography to show that there are ways to be more robust to crossing fiber problems. We've heard about this before the break. And also highlight some of the problems of tractography and the limitation, the precautions, and some of the validation challenges for our field as well. So let's go and talk about tractography and how to get towards optimal tracking. You have to keep in mind that the tractography pipeline is a whole process of processing steps. Today I'll be mostly talking about masking and tracking procedure, algorithms to do that, but keep in mind that image acquisition, image quality, pre-processing your data, correcting for the artifacts, the distortions artifacts, the noise in your data, and accurate local estimation is crucial. So if you want to have a good tracking, you need an optimal processing pipeline. So even though I'm not covering these parts today, keep that in mind because it's crucial. A good tracking algorithm with bad data will produce bad tractography. So let's just briefly mention local estimation. We've heard a lot of nice talks this morning about it. I just want to quickly challenge the audience. If you read the question here, how many voxels do you think, the proportion of voxels where you have crossing fiber problem in diffusion MRI? So at a typical resolution of 2 millimeter isotropic, how often do you think you have crossing fibers in the brain? Less than 30%, 50% of the time, 2/3 to 90%, more than 90%. Turns out that it's between 2/3 to 90% of the voxels that have an underlying crossing fiber configuration. So fiber populations kissing, crossing, branching, bottlenecking, dispersing within the same voxel, leading to a flat tensor profile, but where there should be multiple underlying diffusion orientations. And that's a huge problem. Essentially, what I'm saying here is tractography should not be used with a diffusion tensor or the principal eigenvector of diffusion tensor. So we need to go beyond DTI and be able to handle crossing configurations. And we've heard it this morning before the break. There must be 100 papers on modeling crossing fibers. I did my PhD on this subject. There must be at least 100 techniques to do it. For this talk, let's just assume we can recover a fiber orientation distribution function. That is, at every voxel, a distribution of most likely orientations. And there can be more than one at every voxel. So either as discrete peaks or as a function on the sphere describing the most probable directions of diffusion. Again, highlighting that in these configurations, the diffusion tensor is a flat profile without any preferred direction. Then tractography comes into play. The computational process of connecting the dots is tractography. And so I see my friend in the crowd here. This is a courtesy of Alex Lehman's slide. Just like any kid could do on his drawing book, tractography in a crude sense is nothing more than connecting the dots, following the directions and reconstructing the pathways given by the local voxel orientations. So if you place a couple seeds in the brain and you follow the leader, tractography will reconstruct pathways that are always following the local orientations parallel to the path. So this is an example with 4 seeds in the middle of the corpus callosum leading to 4 streamlines here visualized as tubes. And if you do this across the whole brain, placing seeds in every voxels of the white matter, you can generate these tractograms. So that's the term used for the union of all streamlines produced by a tractography algorithm. Tractogram, defining or modeling or estimating the white matter pathways. But what's going on underneath? What's underneath the hood? How do we get there? What are the computational tests, aspects of it? And as you can imagine, I don't know how many computer scientists or engineers in the room, but there are many parameters hidden from you when you do tractography. You have to choose a step size. You have to choose a stopping criteria. You have to decide if you're allowed to turn 90 degrees or not. All these things come into play, and unfortunately, there's no one set of parameters for a whole brain tractography that's optimal and that's known today. So let me try to just highlight some of these aspects so that you think about this the next time you look at a tractogram or you try to run a tractography yourself. First image here, let's just take a very crude example, a field of orientations, and I'm going to place a tractography seed from here, from the corner. There are many ways to step into this field. You can imagine yourself just drawing a line through this is not that easy. So for a computer or an algorithm to do this, here's 2 different ways to do it, for instance. Either you do nearest neighbor interpolation, and you get this jagged, very discrete-like streamline. Or just by doing linear interpolation, that is when you step outside the center of the voxel, you interpolate with your neighbors, you can see how different the path looks like. They don't finish at the same location, and they don't follow the same path. So that's all happening underneath the hood when you're launching a tractography algorithm. Another very simple example. Imagine this field of orientation, just circles. Extreme case, of course. We don't have streamlines that go in a circle. I hope so. I don't know. But less likely that we have circles, streamlines, or fibers in the brain. But let's imagine we have a seed point here. And look at all the different possibilities if you change step size. So this is the way you step through the grid, your voxel grid, versus interpolation first order, second order, or fourth order. As you can see, the worst case, if you take big steps and use first order integration, you actually not even complete the circle. You get out the grid. So that's a take-home message to keep in mind. If you're trying to track very curving structures, you have to tune your parameters to be able to walk through your grid and be able to capture this high curvature. So typically a recommendation, a good recommendation, and this is debatable, but most of the software these days recommend using 1/10 of the voxel grid as a good step size and using a high order integration steps. And then there's an explosion of techniques to actually connect the dots. You can use deterministic, probabilistic, and many other advanced techniques, which I'm not going to cover today in the interest of times, but a lot of literature on the tractography problem. Let's go just through the basics. The first most intuitive approach is the deterministic tracking. That is, you start, sorry, how do I go back? Okay, you start from a seed and you step in the principal direction. If you come into a voxel where there are two principal directions, you keep going in the same direction that's closest to your incoming direction. That acts as a sort of smoothing, regularization. And if you restart the tracking from the same seed, you get the same result. It's deterministic. You always pick the maximal direction given inside the voxel. In a probabilistic setting, it's different. You use the whole fiber orientation distribution. So what you see here on the left is the act of randomly picking a direction given a fiber ODF representation. So you can see it's sampling around the maxima. So in a sense, this is the way to include a knowledge of uncertainty on your fiber directions, or seeing the voxel as a continuous representation of potential orientations. And then the idea is the same. You need a step size and you need a curvature constraint to walk probabilistically through your orientation field. And so here's a result. A tracking algorithm from the middle of a cingulum bundle. Deterministic tracking can produce something like that. This is something that looks, the ground truth of the cingulum bundle should be a long curving structure like this coming in the temporal lobe. And you can see that deterministic tracking in this setting has missed some of the high curvature area. In a probabilistic tracking, well, you can see at the beginning the uncertainty or the principal fiber direction of the fiber ODF is quite well defined, so very tight, 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 and as you get to the end, and then you get a bit of fanning, your probabilistic track tend to explore the whole structure. And one of the things we can do with probabilistic tracking is turn this into a visitation count. That is, count the number of times each voxel has been traversed. So you can create these heat maps or these connection scores that tell you, given a seed point, what is basically the probability of being connected to this seed point. How about DTI versus Hardy? Well, we've seen it today, but here's a clear example in the Centrum Sumio Valley. This is an area where the corpus callosum crosses the corticospinal tract, and also coming out at you here, there's a superior longitudinal fasciculus. And you can appreciate that here the tensors are flat discs with one principal direction, as opposed to a fiber ODF reconstruction, Hardy reconstruction. This could be a kurtosis ODF, this could be a multi-tensor fit, name it. But essentially having here, you can see that you have two-way and three-way crossing, and then there's hope of tracking through this region in three ways. And so here on the right, on the left, on the right for you is the fiber crossing tractography, handling crossing fibers. You can see here getting most of the projections of the corpus callosum laterally fanning, as opposed to the DTI, which typically just gets the mohawk superior projection U fibers of the corpus callosum, missing out the lateral projections. And also here with crossing fibers, better capturing of the high curvature area where DTI just misses it. Another example here where a DTI tracking seeded from the brainstem will typically just get a very straight cortical spinal track to the feet area, whereas a hardy tracking technique based on crossing fibers will be able to fan out and get the hand, mouth, hand, and feet areas. And then the tractography limitations. As you can imagine, if you're probabilistically tracking or deterministically tracking along these directions, there's a high chance that you might reconstruct things that look aberrant. How often does this happen? We don't really know. But we know that it's easy when you start looking at a tractogram to find all these spurious or what we call invalid connections. What do I mean by invalid connection? I mean by tracks, for instance, streamlines that stop right in the ventricles. Yes, there's a deep structure nearby, but you can really see streamlines stopping and ending in the ventricles, or ending right in the middle of the white matter, or connecting regions where we don't really know from atlases or from monkey anatomy if these connections exist or not. And these are the most problematic ones. We don't know, does this streamline, does this bundle exist or not? And so how do we validate tractography? How do we validate these streamlines that we reconstruct? So my lab and with collaborators internationally, we've been developing the tractometer platform, tractometer, to evaluate tractography. And so with phantoms, ex vivo phantoms, simulated phantoms, we've started building geometries with known ground truth. I agree with you, this looks quite simplistic, but we started adding complexity like partial volume effects, straight, crossing fibers, short U fibers. And here we can generate a diffusion signal at different B values or single B values to mimic clinical applications and really focus on what the tractography output is in terms of connectivity. Can we quantify what are the good streamlines and the bad streamlines that we find? And recently, last year actually at the ISMRM, there was the first international tractography challenge where we generated an even more realistic phantom that looks like a brain. You'll tell me not exactly, but that has realistic artifacts, motion artifacts, EPI artifacts, that comes from a high-resolution HCP dataset with 25 manually labeled bundles where a T1 and a diffusion signal was generated. So we can actually now run our best tractography algorithm at this and start quantifying what are the valid or invalid connections that we get. So I have a talk later on this week to summarize results from this. And not wanting to be pessimistic, but the news are not so great for tractography. So this is probably one of the striking results or discouraging results is that a lot of these streamlines produced by tractography are actually invalid. They stop too near, they stop in the white matter, they stop in the ventricles, or they don't connect valid endpoint regions. So that's a huge percentage. It depends on the data, it depends on the model, but they're there and they're important. They're polluting our tractograms. And one of the more practical conclusion that you can get is if you choose a probabilistic tracking, yes, you will recover more of the spatial extent of your bundles. You'll be more capturing the variability in your data, but at the price of generating more spurious tracks, more invalid connections, which makes sense. You're exploring the structure a little bit more while you're paying the price for it. You're generating streamlines that are actually invalid. So is there hope? How can we reduce these invalid connections? There is hope. There is a lot of hope. I hope to convince you of that. But so one of the big problem of tractography is the biases to these four things. Position. Tractography is harder in the deep structure and near the cortex than in the middle of the corpus callosum. So there's a spatial dependence. Tractography is easy in some location and it's hard in other locations. Yet our parameters are set for the whole brain. Shape is important. Big bundles, curving bundles, straight bundles, or short curving bundles are different difficulty. It's harder to track a structure like this than a whole, like a straight structure like this. So let me introduce you to one of these seeding bias. Imagine you're placing seeds everywhere in the white matter. Everywhere, everywhere. I'm not going to continue like that, but you can imagine, right? Every such seed is generating streamlines. What do you think will happen? Big, large, easy bundles will be overrepresented. It's quite easy to imagine. You're going to be overseeding the corpus callosum, overseeding this corticospinal tract, and these big, easy tracts will be overrepresented in your tractogram. So be careful, and I encourage everybody that's going to be using tractography to read this paper. Fiber count, integrity, and other fallacies, the do's and don'ts of diffusion MRI. So here's another toy example. Imagine these three white matter bundles. You need to be a bit focused to understand this one. You have this bundle, you have twice as long and 3 times as long the same bundle. Now imagine again, same as the previous slide, I'm putting a seed in every voxel of the white matter. Every voxel here, and I'm launching tracks. If I count tracks afterwards, you can easily imagine that the bundle that's 3 times longer will have 3 times more tracks inside. Right? If this track has 100 streamlines, this will have 200 streamlines, this will have 300 streamlines. Just because I seeded the whole white matter. So that's an important bias. This tells you that if you're counting tracks or if you're producing track density imaging, this is happening to you. So one quick fix is to seed the interface. Imagine the same configuration here, but all we seed is the white matter, gray matter interface here, these red voxels. So if you put just seeds here, you can imagine that all these 3 bundles will have actually the same track count. Just by changing the way you seed it. I'm not saying that's solving all the issues of tractography, but this is reducing the issue of large, easy, long tracks that are overrepresented. So the group of MR-Trix with Robert Smith, my group in Sherbrooke with Gabriel Girard, we've introduced or we suggest to seed from the white matter, gray matter interface to reduce this bias. So since you often have the T1 weighted image, we have the tools nowadays to extract this interface and to better seed from the interface. Another way to include anatomical priors is to inject the partial volume maps from a T1 segmentation. So if you know your tissue classes, gray matter, white matter, CSF, you can actually forbid streamlines from ending in the ventricles and force streamlines to connect subcortical or cortical gray matter. And one way to do this is to use a particle filter. Here you see in green the fibers or streamlines surviving as you walk through thin structures. So suddenly using a particle filter, which is used by robots to navigate in space, you can better represent the white matter, penetrate into the gyri, navigate through tight white matter structures, and reduce some of the tractography biases. And so I recommend reading this paper, and there's some tractometer validation and some parameter tuning, suggestion, simulation, and comparison with MR-Trix that one might be interested in looking at. How about microstructure? We've just heard about microstructure. We have new fancy multi-B value acquisition. Can microstructure help us reduce the number of invalid connections? Can we be better at figuring out the crossing fiber problem? It turns out that there might be hope, because as we saw in the previous talks, let's say here is a simulated signal for calibers, axon, tubes of 8 micron diameter and 4 micron diameter, and you can see that the diffusion propagator that it produces is different. It leaves a signature. So there might be hope to not only track following orientations, but also track while following local caliber estimation. And this is not ready for prime time. This is just a proof of concept that's been published in MicroTrack and AxTrack from my group. But let's just use this kissing fiber simulator. We use this kissing fiber, and you can easily appreciate, if you see here from everywhere, that you will reconstruct the two branches of the kissing. But it's quite easy to reconstruct the invalid connections, the wrong branches of the kissing, right? And what's scary is if you do this without just taking the orientations into account, you get about 50% valid connections and 50% invalid connections. It's not surprising. Look at the orientation field. How can a tractography know what to do right here? You can get the U, you can get this branch, you can get the curvature branch. So what if we add microstructure? So as I said, AxTrack and MicroTrack from Danny Alexander's group are proof of concept that this might lead to something hopeful. So imagine again the same kissing. Now we generate a Camino simulation where two different axon diameter. One fiber population with a 6.8 micron diameter and one fiber population with a 2.4 micron diameter. We generate the multi-B value diffusion signal for this. We estimate the calibers, the orientation profiles, and here is a visualization of the peaks colored by their local axon diameter, mean axon diameter estimation. So there's a bit of noise in the center here, but you can still appreciate that you can pull these two configurations apart. Your eye is tracking right now. All of your eyes right now are actually reconstructing both fiber groups easily. And so we can run our tractography algorithm on this. Now, adding the constraint, when you have a direction, you want to keep going in the same direction that maximizes the direction, your incoming direction, plus your diameter information. And so you reconstruct the valid connections. You still reconstruct some invalid ones, but now it's only 13% of the streamlines that are actually wrong. Now you've jumped up the percentages to 87%. So it's still challenging. There are still many, difficult crossing information here. But there is hope to reduce this number of invalid connections. So let me end with more ways to reduce invalid connection. And that is with the good old regions of interest. You can use an atlas. You can manually draw regions of interest. That's probably the most powerful way to find what you're looking for, or to remove what you don't want to see. And neuroanatomists and virtual dissectors have been doing this for years now. But it's a way, in a sense, to inject anatomical priors. And I like this new white matter query language here. This is an example of the query produced. I'll remove this if you let me just. This is an example of the sort of query you could ask. The arcuate fasciculus is defined at endpoints in the inferior frontal gyrus left or right, middle frontal gyrus left or right, and precentral gyrus. And you can concatenate a bunch of definitions like that and start dissecting away your white matter using, for instance, a free surfer parcellation. And you can see these bundles are not perfect. They're not free of spurious tracks, but they're pretty clean. So it's a way to impose some anatomical structures on your tractography dissection. I'll skip that. So in conclusion, a few recommendations. Do not use tractography as a black box, first of all. There are a lot of parameters underneath that you might want to tune depending on what you're looking for. Are you looking for a very curving bundle, or you're performing a whole brain tractogram? Think about your parameters. Be careful of how you interpret your results. Your tractograms are full of invalid connections, so keep that in mind. Restrict yourself from counting tracks or using terms such as connection strength. I haven't talked about connectomics here today because the goal was tractography, but you've seen enough of the limitations to say that counting tracks for now is dangerous. And do use anatomical priors, regions of interest, or multimodality if you have them, to start defining bundles and start performing tractometry or studies of tractography bundles. So with that, I think there's limitations of tractography, but there might be a bright future where we can try to fuse local estimation and tissue properties with the regularization and tracking through these orientations and local features. Thanks a lot, and I'm around for questions.